Hello everyone, my name is Walter Ausserer. I'm the Senior Business Unit Manager for In Vivo Pharmacology at the Jackson Laboratory. And today I'm going to be talking about PDX models for oncology research and preclinical prediction. Now in the talk today I hope to communicate sort of three main points. The first of them is how these models are made and why they're made. Secondly, why the research community is so excited about using these models. And then conclude with some examples of work by oncologists and oncology researchers using these models today. Now, in this webinar format, you can submit questions on the Q&A box on your screen. And at the end of the talk, I'll try to get to those as I can. I expect my presentation to last about 30 minutes. And then after that, I'll move on to questions. First, a word or two about the Jackson Laboratory. The laboratory was established a long time ago, about 85 years ago now, and it's located in Bar Harbor, Maine. Now, it was really established to focus on mouse models to understand basic biology and to understand human disease, and it's grown very significantly over the years. Today, we have more than 1,200 employees there. 47 faculty members, and it's a large mouse production facility as well. Also part of our charter is to be an international training center, and that's actually become very important. There are a significant number of Nobel Prize winners who've passed through the laboratory either as high school or summer students, so it's a very significant part of what we do. About 10 years ago, we established a West Coast presence. And that's at the Sacramento, California facility, and that's where I work. This is probably the world's largest facility for producing mice at the highest health status. And it's also where we perform the in vivo pharmacology services and our PDX program. So all the PDX models we'll talk about today are created at the Sacramento facility. Very recently, only a couple weeks ago, we opened a third location in North America, and this is the Jackson Laboratory for Genomic Medicine, and it's on the University of Connecticut Health Sciences campus in Farmington. And this sort of closes the circle for us. We were really focused on mouse genetics earlier on, then on PDX models. We're incorporating both mice and human tissue, and now this facility is really focused completely on human genomics and human genetic medicine. So we're very excited about this new facility. Now, when people think of Jackson Laboratory, they really think of Jack's mice. And that's because the Jackson Laboratory is an NIH-funded resource to provide mouse models to the research community. There are over 7,000 strains of mice that we maintain, and we're shipping this year almost 3,000 mice to the research community. The real cornerstone of our program, though, is this third point here. It's genetic quality, maintaining the highest genetic quality and purity and the highest animal health status. And we are really, truly the leaders worldwide in animal health status. Now let's talk about PDX now. And the whole PDX concept came about because of the problem that's demonstrated in this slide. Today, only about 5% of drugs in our clinical trials actually make it through to approval. And what that tells us is the preclinical models that are used to develop these drugs just aren't doing the job they should be doing. We're losing about 30% of drugs due to attrition because of lack of efficacy. We lose another 30% of drugs due to lack of safety. And this has just created a huge problem for developers of drugs worldwide, and really is the driving force behind the development of new preclinical models. And really the PDX model is among the most exciting of the new generation of preclinical models. And that's really because it does a much better job of predicting how a drug will work in a human system. Now, mouse models have been used as preclinical models for many, many years. And what's shown on this slide is the classic xenograft model using a nude mouse. So if you look on the left of this side, this is a nude mouse that is harboring a tumor. In this case, it's an MCF7 human breast tumor cell line that's been injected into the mouse under the skin and has grown into a sizable tumor that you can see. Now, after treatment, you see the tumor can fully regress and disappear. The problem with these models are really twofold. First one is the need to implant a cell line. These cell lines have been established and grown in plastic for years, sometimes decades, and they really represent a pure cell population that has little relationship to the natural heterogeneity of a tumor in a human body where you have many types of cells, basically that. Now, in terms of the second issue with these, it has to do with the mouse itself. Nude mice are very good at supporting these established cell lines, but their immune system is such they typically will not support the engraftment of more complex tissue, such as real tumor tissue. And if we're going to study the heterogeneity of real tumor tissue, we really need to be studying that tissue itself, not some purified cell line. Now, in order to come up with a mouse that will support the engraftment of human tissue, you've got to step back and look at the immune system. That's what this cartoon does for us. So when I think about the mouse model and the immune system, on the left-hand side, we see the innate immune system, where we have the complement system, the macrophages, and this is sort of your first line of defense in the immune system. And then on the right side, the blue circles, we have the adaptive immune system, things like T lymphocytes and antibodies, and then the coordination of the two systems through the cytokine signaling. So this is a cartoon of an immune system, but when we're thinking about mouse models, you can also think of each of these circles as an obstacle to the effective engraftment of human tissue. And so what we're going to do when we create a mouse model that will accept human tumors is we need to cripple this immune system as much as we can. And this is a project that's been going on for years in our laboratory and many others. But the latest state of the art is something we call the NSG mouse. And its immune system is shown in this cartoon, where we've basically knocked out almost all of the innate immune system except for the granulocytes. We've knocked out almost the entire adaptive immune system except for a little bit of dendritic cell activity, and we've severely crippled the cytokine signaling. So because the mouse is so immune deficient, it will accept a whole variety of different human tissues, including solid tumors and liquid tumors like leukemia, and not reject those. And so this is the so-called NOD-SCID mouse, which is the basis of all the PDX models that I'll discuss today. As shown here, the NOD-SCID mouse, or the NSG mouse, it's deficient in T cells, B cells, natural killer cells, and very importantly, it has a long lifespan. As long as you maintain it in a relatively sterile environment, these mice will live out easily past a year and are very, very healthy, in fact. And so they do support the engraftment of a whole variety of different tumors and cell types. And so everything I'll talk about today now, all the PDX models are based upon this sort of amazing NSG mouse background. Now, when we are creating these PDX models, the most important step is the first. That is getting the human tumor from the clinician, from the operating room, and getting it engrafted into a mouse within 24 hours is our cutoff. And so you can imagine the logistics there are difficult, and we've worked very hard with a number of cancer centers nationwide to put this network in place where we can get the fresh tissue directly from the operating suite, directly to our facility, into the mouse within 24 hours. Now, in our hands, we find that just about half of the tumors that we bring in will engraft successfully into the NSG mouse. It depends somewhat on the stage of the tumor and how aggressive it is, and also the type of tumor. For example, certain types of breast tumors require estradiol, for example, and you need to be able to engraft in a system that has the hormones needed. But in general, we find about 50% as a rule of thumb. Now, once those tubers have demonstrated they will successfully engraft, that triggers us to do our characterization panel. So we do a variety of different genomic characterization techniques, such as gene expression arrays. We do copy number variation analysis. We do a lot of sequencing of specific genes to look for mutations. And then we place all those data into a database. We also then take that tumor, we take part of it and we cryobank it. But we take the other part and we expand it. So typically, the tumor will be then engrafted in what we call the P0 cohort, shown here as 4 mice. Typically, it could be up to 10 mice where we engraft that tumor, and we grow up a large enough volume or mass of that tumor that we can then archive it for future use. Now, these archived tumors then can be used to create any number of different mice. Very often, we work with pharmaceutical companies that are testing drugs or combination of drugs, and we'll perhaps need 100 or 200 mice with the same tumor in that. So we grow those from these donor mice, usually at the P3 or P4 level. That's passage 3 or passage 4. We can grow up to a couple hundred mice, and then those are ready for any type of drug efficacy studies. Now, in terms of the sources of these tumors, as I said, we've established quite a network. I think early on we worked a lot with UC Davis because we are located close to them, but we've expanded now into more than 20 different cancer centers. These cancer centers are sending us directly from the operating room both liquid and solid tumors, and along with those we get a lot of the de-identified patient identification, such as the age of the patient, the treatment history, et cetera. We also, when we can, we do receive the histology of it, so we can compare the histology of the patient tumor to the tumor once it's been in the mouse. But typically, in terms of experimental work, these are used for compound efficacy studies. That is, testing single agents or combination agents in a very defined tumor with very defined genetic characteristics. I will say that many of these studies take place actually at our CLIA lab in Sacramento, where we have a variety of surgeons and PhD scientists that support this activity. Now, where we are today, this is a little out of date, but we have somewhere between 350 and 400 of these PDX models that we have validated, characterized, archived, and established for use by the research community. You see, it really varies the type of tumors. We have bladder tumors, brain tumors, a few endometrial tumors, et cetera. But our goal here is to keep building and working with the research community to have a really comprehensive resource of different tumor types with different genetic characteristics. And all those data that we collect then, we make available freely, with free access to the research community. And this is one of the areas that really sets the Jackson Laboratory apart from other other suppliers of PDX models. Is everything we have, we make freely available to the public in something called the mouse tumor database. This is through access through the Jackson website. And there's an area, you can just click on PDX tumor search, and you can search the entire database by the tumor identifier, the primary tumor site, for example, being a lung tumor, the diagnosis, and very importantly, the gene variant. So if you're looking for a specific mutation that a therapy is targeted to, this database allows you to find multiple models typically with a specific genetic mutation for those drug testing studies. So if I click on, in this case, a bladder tumor, for example, you see what comes up here is an entire list of different tumors that are available in the database with patient information and also some information on the genetic makeup of the tumor. If I pick one of these and click on it, and we dig down deeper and really see what's available for each of these tumors. That is, there's patient information, there are copy number variation, gene variant information, growth characteristics, and all the genomic information we have. So again, you can just go publicly available website at Jackson Laboratory and review all this information. Also connected with each tumor, we have what's shown here is the gene expression. It's called the Z number, whether it's an overexpressor or underexpressor of specific genes. And we also make available something called copy number variation that looks at the representation of specific genes in the genome, whether there are more copies than normal or fewer copies than normal. Another aspect of our program here, which is quite unique, is the way that researchers can access these models. We provide, and we're really one of the only laboratories that provide what are called study-ready cohorts. That is, these are mice that we have engrafted with tumors, characterized, and we will ship to any researcher that allow them to do drug efficacy studies or other basic biology studies. So any of the solid tumors we have in the tumor database, we have some AML models that are very popular. That's acute myeloid leukemia. These are engrafted with myeloid leukemia cells that are isolated from individual patients and are used to develop anti-leukemia drugs. And we also have a very popular line data called humanized NSG mice. These are mice that have been engrafted with human hematopoietic stem cells and develop human immune systems for infectious disease research, sort of a sister product line. In addition to providing these study-ready cohorts to investigators, we also can do drug efficacy testing at our CLIA facility here in Sacramento, and we do quite a volume of business working with biotech and pharmaceutical companies and some academics as well to test new drugs. And we have a new program that's shown here, the third thing, called PDX Live. And this is a way of taking the most popular tumors and making them available to researchers quickly. One of the traditional problems with PDX and models is it takes quite a while to take a tumor from the freezer, thaw it, and graft it, and grow up a cohort. It can take several months to generate the mice needed for a study. And with PDX Live, what we've done is taken the most popular tumors, for example, some of the breast, colon, and lung tumors that are very widely used, and we maintain those mice on the shelf. And so very often you can get the cohorts of mice within a week, days, weeks, or maybe at most a few weeks, not having to wait for the months that it traditionally took. Now, in terms of areas where we're strong, I think one of the big areas is the non-small cell lung cancer. What was shown here is a panel of the non-small cell lung cancer models that we routinely work with and make available. And you see we have a lot of information about the patient background and the genetic makeup of the tumor and the genetic makeup of the PDX. And basically, most of the driver genetic mutations that are of interest in research are available from our resource. We've also taken pains to be deep in this area of triple-negative breast cancer. When I say triple-negative, that means it's estrogen receptor negative, progesterone receptor negative, and HER2/neu receptor negative. And this is important clinically because these tumors are troublesome to treat, typically have to be attacked with combination therapy, and are a very active area of research for new therapeutics. And so we have established this panel here. We're going through a lot of the standard of care drug studies on these now and looking to expand this even further. Now, I mentioned the estrogen receptor negative breast cancers on the last slide. What's shown here are a couple of receptor positive tumors. On the left, we see an estrogen receptor positive tumor, and on the right, a progesterone receptor positive tumor. The importance here is these estrogen receptor, for example, positive tumors typically would not grow in mouse models because they're dependent on human estrogen for their growth. And a lot of the previous mouse models would not tolerate the presence of the human estrogen. They would develop crystals in their bladder, et cetera. Now, we found that the NSG mouse is very good at tolerating the presence of estrogen. And in fact, we typically implant a small capsule of estrogen in the neck of the mouse, and it does support the growth of these estrogen receptor positive tumors, a very sort of unique niche for this model. I want to talk now about the structural fidelity of these tumors, because it's really quite fascinating and remarkable, I think. What's shown here are some histology slides from a colon adenocarcinoma. And this is a KRAS mutation, a very common driver gene in colon and other types of carcinoma. On the left slide or photo, you see the patient tumor histology. In the middle, you see the P0, or first passage of the tumor through a mouse, histology. And secondly, the P1, the second passage of the tumor. And what's remarkable, you'll see here, are these crypt-like structures that are very indicative of the adenocarcinoma type of histology. So you see that these structures developed with remarkable fidelity at multiple passages in these PDX models. Also shown here are two histology sections of a colon adenocarcinoma from a metastatic growth that is taken from the lung. And we see on the left, this is the patient histology. On the right, we see after the first passage through a mouse. And again, the characteristic glandular structures that you'd expect to see in an adenocarcinoma are very faithfully recapitulated in the mouse model. Sticking with adenocarcinoma, this is an example of a non-small cell lung adenocarcinoma. And the point here is really to focus on the gene expression data. We see the histology here of the patient tumor, the passage 0, the passage 1, and the passage 2, with the recapitulation of, again, the glandular type of histology. But this tumor, the gene expression data shows first that it's a KRAS mutant, so KRAS is probably a driver gene in this, and it's shown to be EGFR wild type. And the importance there is there are a variety of different therapeutic agents that target mutant EGFR. And so if we look at this tumor, our patient, we'd expect for those therapeutics that target mutant EGFR not to work. So things like erlotinib. And in fact, when we test this tumor, that's exactly what we see. What's shown here in the black line is the vehicle control, and this is the growth curve. What's shown in the blue line is that same tumor treated with erlotinib, which targets the mutant EGFR. We see essentially no impact of that drug because this tumor does not have the mutation. But when treated with cisplatin, is the red line, we see that it does respond. So the point here is these PDX tumors have been shown to be really remarkably good at predicting therapeutic outcome and which drugs will work and which drugs won't. At recent conferences I've been to, the number is typically that for the older models, such as the Nude mice with cell lines, the predictive power is only about 30%. So it's not that much better than random sometimes. But these PDX models, often we're seeing greater than 90% predictive power for the use of drugs. And this is really what's gotten the oncology community very, very excited about these models. The other very exciting thing about these models is what you can do with them in terms of studying different drugs. You can really evaluate everything that's available by creating enough mice and designing the right study, things you could never even dream of doing in a human setting. So you can rank order the efficacy of single agents. Very exciting, you can study different drug combinations. Not only drug combinations, but the timing, that is, drug A first, followed by drug B, or maybe you'll try drug B first, followed by drug A. And this lets you really kind of probe into the onset of resistance and how the drugs can interplay. So really a fascinating tool for studying the interplay between different drug and dosing regimens for individual tumors. I'm going to change gears now and talk about liquid tumors. And this is an area where the NSG host has been a real superstar. So very often you cannot, these liquid things like leukemias have not taken very good hold in different mouse models. They really work very well with the NSG host. And in these models, what we do is we engraft these mice with leukapheresis samples. So these are samples of cells that are isolated from individual leukemia patients, preserved and then injected into mice to create these models. And the two models that we work most commonly with, one is called a FLT or FLT3-ITD. This is it's basically a growth factor receptor, and this ITD, this internal tandem to repeat, is one of the most common, if not the most common mutation seen in AML patients. Secondly is something called an IDH1 mutant. IDH1 and IDH2, they're actually metabolic enzymes, and when they're mutated like this, they interfere with a normal differentiation of cells. And so the two models I'll talk about today and what we most commonly sell are the FLT3-ITD models and the IDH1 mutant models. To create these is actually quite a simple process. We take the mice, we do a whole body irradiation, which sort of opens up a niche in the cells, opens up a niche in the bone marrow of the mice. We can then inject these human leukemia cells. They will take hold in the bone marrow and other places. And about 14 to 28 weeks in, as we monitor those, we see the human AML cells appearing in the peripheral blood and in other organs as well. This is an example here of where we find these populations of the human AML cells. So on the left side, we see two different models in terms of where the percentage of cells in the blood at different time points. We see the cells in the spleen at different time points and in the bone marrow. So you really do see these cells proliferate as you'd expect a leukemia to do in a human setting. And we also see these models responding to standard of care drugs exactly like you'd expect a human AML patient to respond. In this case, the drug is called Ara-C. It's a standard of care for many AMLs. And we'll see the decrease on the left here of the number of AML cells in the bone marrow of these models. We see a decrease in these cells in the spleen, these models, and also in the peripheral blood. So again, therapeutically, the cells in the mouse model respond exactly as you'd expect the cells to respond in a human model. Now I'm going to conclude now by talking about sort of where the future of this work is going. And it really has to do with, excuse me again, the human immune system and how the human immune system can be modulated. I think the most active area of cancer research right now is human immunomodulatory therapy, where you modulate the human immune system to attack the tumor itself. And what I'll talk about now and conclude are different models that we are moving toward for modeling human immunotherapy. And that begins with something we call the humanized NSG, or in this case, the CD34 plus humanized NSG. What we're going to do here is create a mouse that has the human immune system as a first step. So again, we create these models exactly as we created the human AML models. That is by irradiation of the mouse to create a niche in the bone marrow. In this case, we're going to engraft human hematopoietic stem cells. And then after several weeks, we see the appearance of human B cells, human T cells, and most of the component of human immune cells that you would typically see. There are several different models of this that display different levels of human immune function. The most common is the so-called CD34 humanized mice. These are created, as I just said, by engraftment of human hematopoietic stem cells. These have a full range of human immune cells with some exceptions. But the big thing here, they have a very long, healthy lifespan, many months. And so it can be used for very long studies. The downside of these, they do have somewhat weak B cell function and poor IgG function. So if you're in a system where you need that B cell function or more IgG production, you might consider something that's called the BLT humanized mouse, which is based on the CD34, but has the difference being we do implant small pieces of liver and thymus under the kidney capsule of these mice. And what that does, it helps with the T cell maturation, and we do get improved immune function in these cells for certain things like mucosal immunity. If you're studying HIV infection in the body, you don't see mucosal immunity in the CD34 cells. You do definitely see it in the BLT mice. The downside of having the more souped-up immune system, though, is the mice do not live as long. These typically last about 20 weeks, and they develop a graft versus host type of phenotype. So improved immune system, lifespan a little bit shorter. Also available are something called PBMC humanized mice, where we basically take the NSG mouse and just engraft a full complement of PBMCs from a human directly into the mouse. The upside of these is they do have the full complement of human immune cells, but again, you do have the short lifespan, and after about 30 days, those cells then will turn on the mouse, and you get this graft versus host. So depending on the type of what you're studying, there are a variety of different options here. And all these options are being looked at today in the very early stages to see if these are going to be models for immunotherapy in the future. That is, they're used for infectious disease, as shown on this slide. They're used to study gene therapy and et cetera. But they can really be used to study cancer, we're hoping, because if you can take an individual mouse and graft it both with human immune system cells and with human tumor cells, I think it's going to open some fantastic areas for research. And this is just my concluding slide today, showing that what the areas where these humanized mice are really moving forward. And the third point is what I want to talk about. These human immuno-oncology models is really our focus moving forward, and I think you'll see the field move very aggressively to these new type of PDX models very soon now. So I want to thank you for joining me. I hope I provided a good overview of the field of PDX work, and I'd be very happy to take any questions now submitted through your chat window. And I do have some. Now I figured out. The first question has to do with PDX Live. It says, how long will these tumor models maintain the original traits on the shelf? I think it basically has to do with the stability of these models. With the PDX Live, what we do is we always try to go back to the original tumor, and so we maintain those at early passage. That is typically a passage 3 or passage 4. So as long as we have the stalks of the tumor, they always go back to that early stage that we have found to be optimal for demonstrating the proper heterogeneity and genetic characteristics of a human tumor. Second question. The NSG mice, are there NSG mice that will develop drug resistance? And can they be used to study overcoming resistance? I will say that there are certainly PDX implanted NSG mice that are being used today to study drug resistance. They do actually can develop resistance and can be used to study drug resistance. There are a number of studies going on at UC Davis looking at exactly that. They have a very active study program in bladder cancer, colon cancer, lung cancer, and this is one of the areas that they're investigating today. I'd be happy to take more questions. Those are the two that came in so far. Well, if there are no more questions, I'll sign off. I'll thank you for your attendance today, and feel free to contact me directly if possible. So I'm signing off now. Thank you.